Hello everyone, I'm Mr. Furlong, and today we're going to take a look at changes in the signal transduction pathway. It is possible to change the signal transduction pathway in order to alter a cell's response. Many types of our medicines do just this. So just as a quick review, remember that we have a ligand, that's just a molecule that is going to attach to a receptor. That receptor is embedded in the cell membrane. We have some proteins. These are our G proteins. And we might have something like an enzyme where the ligand attaches to the receptor. It causes the alpha unit on that protein to then cause a conformational change in the enzyme. And then something like ATP might become cyclic AMP, and this is our secondary messenger. Kind of sloppy there, but you remember. So let's take a look at where some changes could happen. First off, I want you to take a careful look at the blue receptor. Watch how that changes here as I move the slide. It has a different shape to it. So it's possible that there is a mutation in the receptor protein, which now has a different shape. So when the ligand attaches to that receptor protein, nothing happens. Hence, nothing beyond the ligand attaching to the receptor is going to happen in this signal transduction pathway. Let's take a look at another example. Look at the orange enzyme. Now watch how it changes real slightly. The mutation is now in the enzyme, so the ligand attached to the receptor, which let's say it fits this time, that alpha subunit will head on over to the enzyme, but because the enzyme doesn't have the right shape, guess what? That ATP, it's not going to become cyclic AMP, CAMP, and everything else farther down in this signal transduction pathway, it's not going to happen. We know that chemicals can interfere with any part along this pathway. Now, when other chemicals are involved, that could inhibit the pathway or it could activate the pathway. And again, we have many types of medicines that will do one of these two things, inhibit a pathway or activate it. So let's take a look at an example of this. Here is a diagram that was on an old AP test, and we see some FGFR proteins. I'll highlight that there for you. And there is a chemical, a ligand, called the fibroblast growth factors. The fibroblast growth factor can attach to those FGFR proteins, causing a conformational change and then activating the signal pathway. So there's an example where a chemical activates the pathway. Another example is aspirin. Aspirin can inhibit a pathway. In one of the many things that aspirin does, it can affect the cyclooxygenase enzymes, which there's two of them. We call them COX-1 and COX-2. Aspirin, through the process of acetylation, will break off a small functional group and it attaches to these enzymes. Now with the COX-1 enzyme, the enzyme becomes inactivated. And so this can actually help with helping the relieving of pain, for instance. And specifically, it's attaching to a serine amino acid on the COX-1. But on COX-2, although it can block pain, it can also activate that enzyme and can cause it to produce productive lipid mediators. So any type of mutation or any type of molecule can either inhibit a signal transduction pathway or activate it. And I'll see you in class.